Okay, in your thesis, two chapters are very important. One is result, the other is discussion. So in result, you should try to mention each and everything that you can about your thesis, okay, about your family, any genotypic, phenotypic data, anything. What is discussion? Discussion is actually like if I'm discussing something with you, if there is something figure like I'm telling you that the gene is SSCR exome 6 blah blah, that's it. This is the figure, this is the result. Discussion is like, because the gene is SSCR, which is already mentioned by Zafar et al. In 2016, and in those family, the family that they had mentioned, they have this is phenotype, like the phenotype of ataxia, medullary jaw, bulbous nose, small head circumference, which is correlating with the phenotype of this family presented in this thesis. Or this is one statement, either you are going to correlate the data or you are going to contradict the data, that whatever I mentioned, that this gene has been mentioned by blah by someone at all in this year, they have presented this phenotype, but which is contradicted to the phenotype presented by this family in a white in the thesis PK0S22, suppose. So you are either going to relate or contradict by giving specific references. That's who at all, in which year, which family, is it a French family, Canadian family, Caucasian family, Asian, African, because everything is mentioned in the gene card or the OMIM, that results are mentioned there. Okay, so you have to mention those here in the discussion, discuss those with your family, compare them. Okay, this is called discussion. So discussion contains a lot of references and your words, that how something is already mentioned in the world and what is your result, how they are going to correlate. So let's see what they have written. This section should include discussion of the data generated by you in the light of the relevant literature. So for this, try to choose the recent literature, and also if there is any old literature, whatever is literature available for your gene, you should mention it. Then comes the data of the other laboratories, whatever is it, European, American, Australian, whatever laboratory work is there, you can compare them, okay, and maybe given only to support your data in case the results are different from the previously published literature. So this is the second scene. If it is different, a student can give possible reasons. So now you can tell some reasons that why my family can be different, that can be different, because you are doing the genetic study. So it's quite obvious that your phenotype can be different from any other Asian, Caucasians, or Canadian, or any other population, because genetics is like population based. So yes, this is possible, and you can easily discuss this thing in your discussion. Okay, if you are, in either case, if your results are common, like they are same phenotypes, or if your phenotypes are quite different from whatever is published already. So this is called discussion. This can take your 2 to 5 pages depending on like how you discuss your result. A good discussion shows that you have really worked hard in writing your thesis. So this gives a very good impression to the examiner and to anyone who checked the thesis. They open and see how you write a discussion, because this portion tells your potential, that how you can write. So the discussion must end with a clearly defined conclusion, and so you must mention like what have you concluded by your result, by the whole world result when you discuss. So what's your conclusion, what do you conclude from it? And it can be 1 2 line, 3 lines, and any future prospect. If you are, now here comes, most of you will ask the question, something is not included in my thesis, this thing is not included. So here you have to mention the future prospect of the subject under investigation, that whatever you have done now, this thing can be further investigated by using, suppose, Sanger sequencing, or by doing the cellular investigation like cell culturing, or any animal model can be designed on it to see the further effect of this gene on a particular phenotype. and so on. Okay, so this is the future aspect. You can write anything, or for the like genetic what we call it, genetic guidelines and counseling. Okay, or maybe for like the neonatal screening, and anything that you think that you studied in the literature, you think this can be the future aspect of your particular thesis, then you can give those one or two aspects, or maybe three.